A mutation is simply a heritable change to the genetic material. That means that the DNA sequence is different from the original or wild type sequence. Every time DNA is replicated, mistakes can occur. Fortunately, cells have proofreading enzymes that fix most of those mistakes before they become permanent. Mutations can also be caused by environmental factors like ultraviolet light from the sun, certain chemicals, tobacco smoke, or radiation. Not every mutation is harmful. Some mutations have no effect at all, some are harmful, and a few can actually be beneficial. Evolution depends on the mutations because mutations create new genetic variation. The first type of mutation we're going to discuss is called a point mutation. A point mutation changes one nucleotide in the DNA sequence. Let's start by looking at the wild type sequence. Wild type simply means the normal sequence before any mutation has occurred. Remember that every 3 nucleotides make one codon, and each codon specifies one amino acid. Because we're changing only one nucleotide, sometimes that change is very small, and sometimes it can completely change the protein that's produced. We're going to look at 3 different outcomes of point mutations: silent mutations, missense mutations, and nonsense mutations. A silent mutations changes one nucleotide, but the amino acid stays exactly the same. Remember when we talked about the genetic code being degenerate? Multiple codons can code for the same amino acid. The DNA is changed, the mRNA is changed, but the amino acid sequence stayed exactly the same. Because the protein stays the same, silent mutations have no effect on the organism. Evolution doesn't care about silent mutations. Now let's look at missense mutations. Missense mutation changes one nucleotide, and that changes one amino acid in the protein. Changing one amino acid doesn't always affect the protein. It depends on which amino acid is changed and where it occurs. Sometimes replacing one amino acid with another that has similar chemical properties doesn't affect the protein very much. Other times, changing even one amino acid dramatically changes how the protein folds and functions. One famous example is sickle cell disease, where a single nucleotide change replaces one amino acid in hemoglobin. That tiny change alters the shape of red blood cells and causes the symptoms of the disease. So one nucleotide really can make a huge difference. Our third type of point mutation is called a nonsense mutation. Instead of changing one amino acid into another, the mutation changes a codon into a stop codon. Remember that stop codons tell the ribosomes to stop translating the protein. If a translation stops too early, the protein is incomplete. Imagine trying to build a bicycle but stopping halfway through. You'd end up with something that doesn't work. That's often what happens with nonsense mutations. They usually produce a shortened protein that have reduced function or no function at all. Now we're going to look at mutations that are usually much more serious. These are called insertions and deletions, often shortened to indels. Instead of replacing one nucleotide, this type of mutation adds or removes nucleotides. Remember that the ribosome reads mRNA 3 nucleotides at a time. Think of a sentence that looks like this. Now let's separate that into groups of 3 letters. If I delete the first letter, every word after that point changes. That's exactly what happens during a frame shift mutation. Adding or removing one nucleotide shifts the reading frame, changing every codon downstream of the mutation. As you can see in this figure, the normal sequence, or the wild type, produces a protein. After an insertion, every codon after the mutation changes, leading to a completely different amino acid sequence until the ribosome eventually encounters a stop codon. Because so much of the protein changes, frame shift mutations are often much more damaging than point mutations. One important exception is if nucleotides are inserted or deleted in multiples of 3. Since codons contain 3 nucleotides, adding or removing 3 nucleotides preserves the reading frame. The protein gains or loses 1 amino acid, but the rest of the sequence remains unchanged. To wrap up, a mutation is a heritable change in DNA. Point mutations affect 1 nucleotide and include silent, missense, and nonsense mutations. Insertions or deletions, called indels, can cause frameshift mutations, which change every codon after the mutation. Most mutations are neutral, some are harmful, and a few are beneficial. Without mutations, there would be no genetic variation and no evolution.